Our last speaker of the day is Dr. Joan Massague. He actually used to work in Massachusetts here at UMass Worcester. He moved down to Memorial Sloan Kettering many years ago. Since he's been there, he really has been the world leader in TGF-beta signaling. And of late, he's become much more interested in the mechanisms that enable cancer cells to adapt to diverse sites of dissemination. He just was, at the risk of embarrassing him, awarded a large prize in Barcelona, a large prestigious prize for his research, and then jumped on a plane, truncated his visit to Spain in order to make it here to this symposium, for which we're very grateful. He just ran in from the airport, and his lecture is the Biogen IDEC lecture. It's a way of our expressing our thanks to the leadership role that Biogen IDEC has taken in supporting this symposium. Joan.
Thank you, Bob, but it wasn't Barcelona, it was Madrid, which is even better, especially for a Catalan. It's a pleasure to be here at last, to join you, and to close with drawing your attention to some of the very issues that Klaus Plantel has been bringing to our attention, issues that have to do with what cells do after they leave a tumor and before they engage in explosive metastatic growth. This is, in fact, what clinicians and patients are confronted in most instances when a tumor has been diagnosed, has been removed, and if metastasis will occur, in many cases, is going to occur from what the surgeon has no access to, which is all the already infiltrated tumor mass. Anyway, so this also has to do with the problem of breast cancer, which is where this biological phenomenon occurs. And this doesn't apply, of course, to other types of cancers, for example, lung cancer. In the case of breast cancer, as you've seen, as you know, the tumors very early on can disseminate, can release cells before they were diagnosed and removed. These cells, many of them will die in circulation, but enough of them will infiltrate tissues, most easily, probably the bone marrow, because the sinusoids are fenestrated. And these cells then will fall in a state that we call dormancy, and that we've been very interested in knowing exactly what it is composed of. The implication of this is that while these cells were competent to infiltrate at least some organs, at least the bone marrow, there's evidence also for early dissemination to the lung as well, they were not fully competent to colonize, because if they were, they would have done it, as it happens in the case, in most cases, of lung adenocarcinoma or pancreatic carcinoma. Further implications of these are that if the completion of the conditions necessary for full explosive growth overtaking the host organ takes so long, this further evolution, further progression, must be happening, at least in part, in the host environment, giving rise to thoughts of tumor cells, disseminated tumor cells, evolving into metastatic subspecies, much like different finches evolve into different species in different Galapagos Islands. So my lab, as some of you know, has been interested in testing this hypothesis, in learning about what are the functions that enable these cells to enter and eventually take over these various tumors. We have paid attention to the fact that the first condition for metastasis, once the cells are in circulation, infiltration, is already going to be selecting for different capacities. In the bone marrow, a lower threshold, because, as I said, these are fenestrated capillaries. In the lung, they are not fenestrated. That is more difficult, more challenging. These cells will have to bring in something that they already developed, presumably in the primary tumor, to extravasate, let alone in the brain, where the blood-brain barrier provides an even thicker barrier. And then, of course, the parenchymas that they are confronted with and the milieu of factors and of cellular components will be very different in this long, up to decades in some cases, process of selection for the full competence. This hypothesis we've tested by taking cells from a patient that has, at this advanced stage, metastases in various sites, and presumably, according to the hypothesis, different species of metastases, releasing cells now into the circulation in turn and accumulating in body fluids, including this one, which is available clinically. And using this as a sorter, we can try and separate from this mix cells that are highly competent to form metastases in those various sites, brain, lung, and bone, that mirror where the disease has thrived in the patient. And this was accomplished, as you can see in these experiments. One can take these cells from the corresponding sites, inoculate them into the next set of mice, and see that those that were enriched for, selected for in the mouse or sorted for the bone, preferentially go to bone, lung to lung, brain to brain, and so forth. And then also being able to go back to the unsorted mix and find indeed here the tracers, the fingerprints, the gene expression profiles in individual clones that are characteristic of what was sorted through this pass. And through this approach, a number of colleagues in the lab, starting with Yibing Kang, took to interrogate these cells and ask, what is that a cell that is highly metastatic for the bone, for example, compared to those that are not, or metastatic more for the lung, has at the level of gene expression, at the level of microRNA expression, at the level of whatever you want expression. Yibing did that, keeping in mind that whatever these cells had to explain eventually what is that they are doing as metastatic cells, aggressive metastatic cells in the bone marrow, which is to engage the osteoclasts to produce osteolytic bone metastases. The osteoclasts are the ones that will be resorbing the bone, will have to be rallied, differentiated from progenitors, and they will have to then, in doing so, give rise to a vicious cycle that others, Greg Mundy in particular, had previously noted and proposed, whereby factors released from the bone matrix, BMPs, TGF-beta, IGFs, further stimulate the cells to produce the signals to keep this going. And indeed, when he interrogated by gene expression profile, Yibing Kang, the cells that were highly metastatic to the bone, he immediately found a list of genes whose expression was significantly, provocatively increased. The list is much longer than this. This is just the top scoring ones. And several points were obvious. First, that this list contained items that were already at the time being identified independently by others as important mediators of a cell's ability to thrive and expand in the bone, in the bone marrow and into the bone matrix. CXCR4, the receptor for SDF1, alias CXCL12, which provides, which is thought to provide the cells with perhaps chemotaxis as well as survival abilities. Interleukin-11, CTGF, and others that are implicated by the work of others, and to some extent ours, in osteoclastogenesis. The release of TGF-beta inducing stimulation of these cells induces the expression of some of these genes identified in a TGF-beta independent manner. Interleukin-11 and CTGF are robustly induced by TGF-beta perpetuating the cycle. We went on with this approach to look at lung metastases abilities. This is how a circulating tumor cell sees the lung capillary wall, a wall of tightly juxtaposed endothelial cells. This is how this cell would like to see it. So indeed, for these cells to infiltrate the bone one way or another, they will have to achieve separation and intercalation across cells on the endothelium. And indeed, the list of genes that came out, 54 of a similar analysis done by Andy Min and Gaurav Gupta on cells that were highly metastatic to the lung from the very same patient that I've sampled from which we had isolated the bone metastatic ones, were characterized by these genes, a long list. These are the ones that made the cut. And something that helped at the time came out of asking the question whether some of these genes might already be expressed in primary tumors from patients in a manner that might possibly correlate with the propensity of these tumors to prime cells to metastasize to the lung, suggesting that cells expressing these genes might be primed to form lung metastases. And indeed, of these 18 turn red on the slide, because these are the 18 whose expression in primary tumors was correlated clinically with lung metastases, lung relapse, outcome. These genes turn into a classifier can indeed pick a higher, significantly higher propensity of tumors that score positive, especially triple negative or ER negative tumors. Scoring positive for the LMS are indeed at higher, associated with higher incidence of lung metastases. Biologically, however, the concept was that these genes expressed already in the primary tumors may well be priming these cells emerging from the tumor for infiltration in the lung, the first steps. Not necessarily colonization, because that is something that presumably will be selected for over many years. If such functions were already present, those cells would once again be able to go all the way from the outset. And indeed, analysis of several of these genes by knockdown, singly or in combination, or by overexpression in cells that either highly expressed them and were highly metastatic to the lung, or were not expressing and were poorly metastatic to the lung, went to show that these notions had some merit, were correct, in that several of these genes help these cells pass through an endothelial barrier. Koch2, the prostaglandin synthase that will allow these cells to release prostaglandins, and prostaglandins are inflammatory mediators that will induce vascular permeability. Epiregulin, a ligand for the EGF receptor, acting on cells, cancer cells, among other effects on cell motility and cell invasiveness. MMP1, a collagenase. Angiopoietin-like 4. One might ask, what are these genes doing in the primary tumor? Are these just general aggressiveness genes? Well, they are evidently, if they are expressed there, some of them at least may be selected for providing some useful functions to the cells. But they are not necessarily the very same functions that are being used by these cells at destination, because these genes are helping these cells infiltrate the lung, but not necessarily other sites. Indeed, we went on to find that COX-2 and epiregulin help in the assembly of neovasculature in a tumor, whereas here, perhaps touching on similar buttons, but what they achieve is dynamic interactions between the endothelial cells and perhaps the pericytes that the tumor cells trapped in the lung capillaries take advantage of to pass through. Others, however, may well be perfect bystanders. Angiopoietin-like 4. Angiopoietin-like 4 is expressed in primary tumors as a consequence, one of 100 or so consequences, target genes for one of the factors often present in the microenvironment of a breast tumor and other tumors, TGF-beta. TGF-beta is released by cells in the tumor stroma, myeloid progenitors, the tumor cells themselves, and induces, of course, goes on to act on the tumor cells, included to induce the expression of many genes as a consequence of the fact that TGF-beta is there. Well, of those 100 or so, we go on to find that David Padwa's work, that these cells will be happy to use it, not here. Knocking it down doesn't make any difference that we can tell. It's a perfect bystander, but very happy to resort to it at this stage, because what angiopoietin-like 4 released by these cells as a consequence of having just seen TGF-beta in the tumor stroma before they would move into the circulation and a while later be lodging into the lung, what this angiopoietin-like 4 released by these cells will do to endothelial cells that are contacted by it is it will blow away the cell-cell junctions, something that, once again, is going to facilitate the passage, the infiltrative passage of cancer cells into the lung parenchyma, will increase the numbers day after day of cells that this patient is going to be harboring in the lung, thereby increasing the numbers that the probabilities that some of these cells will go all the way. That is the hypothesis based on these biological observations. Similar approaches recently described and applied to the other major site and the gravest site of metastases, brain metastases, a growing problem, have yielded results that relate to this train of thought. For example, cells that are highly metastatic to the brain and that give rise to metastases that by MRI recapitulate all the features of edema and hemorrhagic core, diffuse distribution in the brain parenchyma that pathologists will see in typical breast cancer metastases in humans, subject to the analysis of what is that these cells have that allow them to pass through this most difficult barrier, the blood-brain barrier, what we find is perhaps not so surprising in retrospect. We find that these cells are happy to use what their cousins learned to use, were selected for being able to use to penetrate the capillary walls for lung metastases. Koch2 comes up. Epiregulin HB-EGF. Interestingly, the preferred EGF family member is the heparin-binding one in this case. This is in samples from 2 different patients subject to this selection analysis and independently analyzed. Angiopoietin-like 4 and others again. But then there has to be something extra, something that cells do not need for extravasation into the lung parenchyma, but they have to to pass through this most demanding barrier. There are going to be, we believe, a number of genes whose products allow these cells to accomplish that. The one that took the lead in our studies is the sialyl-transferase with this unpronounceable name, ST6 for short, which is an enzyme in the Golgi that decorates the surface of the cell with sialic acid, and that with this allows these cells somehow, mechanism under investigator, to pass through blood-brain barrier barriers, both in vivo for brain metastases and also in vitro in blood-brain barrier transwell assays. Well, so all of this can be recapitulated in this sort of cartoon, beginning to put labels along that path that was in the first slide. Genes that these cells must already carry as they leave the tumor to confront the first demand. You pass through the barrier, or else you will be in circulation until cleared. And these are some of the ones that I have mentioned, and some extra ones for the brain. Genes that have been collected later, presumably later in these cells, we don't find so many, in fact, any, associated with passage through the bone capillaries. Doesn't mean they are not there, but it's certainly a less selective barrier. But several have come up that are involved in producing very aggressive metastatic growths, outgrowths in the bone. Also, when we look for, we did find genes that are non-organ selective. These genes, what we look for in this case, were microRNAs that might be associated with metastatic process, work of Sohail Tavazoy, pointing at these 2 microRNAs as negative regulators of the process, microRNA 1 to 6 as an inhibitor of cell proliferation, microRNA 335 not as an inhibitor of cell proliferation. In fact, microRNA 335 is targeting, among others, a short list of metastasis-relevant targets. The most prominent are these 2 so far in our hands, tenascin C and SOX4, and work in progress with Sohail Tavazoy, who is currently now running his independent lab at Rockefeller University, indicates that these genes are providing stem cell niche functions and invasiveness to the very cells that are expressing them. Something that, of course, is very important, very likely to be very useful biologically for these cells to take hold once they enter whatever site. Well, these are events at the beginning and at the end, but most patients deal with these when they come out of the OR. And so that inspired us to begin to try and interrogate what is that tumors must provide to their disseminated cells for these cells to be able to survive. If they are not able to engage in explosive growth, at least they have to be able to survive for a long time on arrival. Pathways, functions, genes that confer that survival after dissemination are buying the cells a ticket for them to go through that evolution that eventually may end in a fully equipped, highly aggressive metastatic entity. That was our reasoning. This is in cartoon form. Before they form those metastases with that cocktail of genes and factors released and so on, these cells arrive there and are confronted by a very different environment from what they are used. How do they survive? So that eventually we'll do this. The approach that we took was to interrogate breast tumors for pathways that are known to support survival in one way or another, and ask whether any of these bona fide cell survival pathways, their expression in primary tumors might correlate with the propensity for these tumors to relapse, especially to relapse late, to relapse after many years, meaning that they have to be really good at surviving while they were not yet good enough at engaging in explosive growth. We did this bioinformatically by deriving, that is the work of Sean Zhang, deriving a number of classifiers that were derived from the gene expression profiles of pathways, such as the RAS pathway, Wnt pathway, TGF-beta, MYC, E2F, and the SRC pathway. These taking cues from the work of Joan Evans of not too long ago, using variants of the pathway gene expression profiles that he derived, and also deriving some of our own, Wnt as well as TGF-beta. Sean interrogated primary tumors, asking the question whether gene signatures, or rather classifiers derived from these signatures, might correlate the expression in the primary tumor with late onset metastasis. Before I show you the data that support these conclusions here, let me familiarize you, in case you are not, with the general behavior of breast cancers classified by the most basic form of classification, ER status, the behavior with regards to metastasis. Typically, the ER-negative tumors are more aggressive. They develop metastases sooner, those patients, but then they plateau. After 5 years, there is a very high chance that those who did not relapse within 5 years will never relapse. Whereas the ER-positive tumors that are regarded as less aggressive, they certainly take longer, on average, to relapse, but they keep going and going and going. And in this particular cohort, it looks like it plateaus, but in others, keep on dropping. These with regards to the overall rate of relapse. When we look at bone relapse, also this well established by others, just done with a cohort of 615 tumors that we resorted to, typically ER-negative tumors have less preponderance of bone relapse, whereas the ER-positive ones are very good, most prominently relapsing in the bones. So classifying a cohort by ER status is going to, in every case, separate, and classically does separate, the curves with ER status tracking with bone metastases. When we applied these various algorithms, one and only one, the one derived from overexpression SRC in cells, gave a classification of this kind. The others did not classify in terms of metastatic propensity relapse in any organ. In the case of SRC, there was this remarkable separation into two groups, two subtypes, SRC signature positive and negative. The positive ones having indeed a higher incidence of bone metastasis, and also, if you notice the curves, a high incidence of late events, well past the 5-year mark, whereas the negative ones did relapse initially, but then they plateau. Well, because there is a correlation, these are the 615 tumors, a non-correlation between ER status and SRC from the literature, that didn't mean much with regards to the ER-positive tumors in terms of whether SRC activity in the cell was cause, consequence, or coincidence of ER status, which in turn is linked to bone relapse. However, this group here was very useful. These are ER-negative tumors, verified to be ER-negative. They are not misclassified, that are scored as SRC signature positive. As you can see, subjectively, there is a higher congregation of density of cases of bone relapse within this subgroup than there is in the ER-negative, SRC-negative subgroup. And similar, indeed, to this density. And these are Kaplan-Meier curves showing that the ER-negative, SRC-negative, few and early bone metastases, the ER-negative, SRC-positive, very similar, tracking very much with the classical ER positive, which are also Src positive. So this then was very useful to us experimentally to separate, dissociate Src from ER status and begin to test functionally whether Src is indeed providing some advantage to these cells. We tested this in two systems. Here is looking at highly aggressive bone metastatic cells are inoculated into the circulation, allowed to form osteolytic metastases, seen here by histology, X-ray, and by fluorescence, luminescence emission with luciferase. If Sean knocks down Src, these plummets, this bone metastatic ability, and if he rescues the knockdown with a Src RNAi-resistant vector, it brings back metastasis, osteolytic metastasis with a vengeance. This has nothing to do with the cells being better at infiltrating. Src, of course, has strong abilities to promote the migration and infiltrative capacities of a cell. That certainly would help, but here that is not rate limiting. What is rate limiting is what the cells have to do once they get in. How do we know that? Because in this experiment, the cells were directly injected into the marrow, and we still see a major difference between the cells that have the endogenous level of Src, which, by the way, in bone metastatic cells, we find it to be higher than in lung metastatic cells, and cells, the same cells that have the knockdown. Again, an order of magnitude drop in the ability of these cells to take off. What are these cells suffering from when they don't have Src? Apoptosis, higher levels of tunnel positive cells in the tumors that do form at a much slower rate in the Src knockdown, or in this case, inhibiting Src, not with an RNAi, but with a drug, one of several that are in clinical trials as Src kinase inhibitors, the dasatinib. Proliferation rates are the same. Rates of osteoclast mobilization are the same. And by the way, these are effects of Src on the tumor cells, of Src inhibitor on the tumor cells, Src family member. That is the Src itself, not other family members. This we know by rescuing these cells after a knockdown with a dasatinib sensitive or a dasatinib resistant version of Src. And the dasatinib resistant, and only that one, confers the ability of these cells to form metastases even in the presence of dasatinib. And Todd Golub has used the same approach recently in his own studies. This is another system, one that is indolent. That one was aggressive. This one is derived from another patient, grows very slowly in the bone at this level of cell inoculation. Even more slowly, we inoculate fewer cells. After a month and a half, we can detect no signal whatsoever by luciferase, which is a very sensitive assay. When we allow signal and growth to be accrued by these cells, again, we do see that Src knockdown diminishes this, and Src rescue brings it back. When we resort to the levels of cells, inoculated levels of cells, that will give rise to no discernible metastasis by any technique, and we flush the marrow from these mice after 5 weeks, and count colonies of cells that survived, that were sitting there by all intents and purposes in this latent state. Do they grow as metastases? Maybe, maybe not, but clinically that would still be called latent disease because you can detect it. Well, again, Src knockdown decreases very much the content of such cells in the bone marrow. Well, then Src is preparing these cells to thrive, to survive in the bone marrow before they are able to have the full complement of functions necessary for an aggressive overtake. That in turn suggested to us that Src might be preparing these cells to respond well to something that is available in the bone marrow, because this advantage that Src provides was not seen in cells, even in the stem cells or in lung metastatic cells, allowed to nest in the lung. No difference there. So something in the bone marrow and bone metastasis microenvironment might be available to these cells, and these cells might be able to use it provided that they had a robust level of Src function acquired, however. To identify what that might be, we analyzed the expression of secreted factors in metastasis tissue from patients and memorial. Some 50-some samples were scrutinized by gene expression analysis to find this set of genes whose expression was elevated in bone metastasis samples, but not in the others, in any of the others, or only in some of the others, some of them. Several of these were already known to be present in bone, bone marrow, bone matrix, and in osteolytic process. Two are highlighted because of all the ones that we screened, these two, and only these two, required SARC to provide cell survival functions to these cells. Here's looking at the ability of the cells to respond to CXCL12, alias SDF1, with AKT activation, with survival in limiting serum conditions. This ability is deprived if we knock down SARC, brought back if we put back SARC. So SARC is functioning as an enabler or enhancer of the ability of these cells to respond to CXCL12 via the CXCR4 receptor to provide survival functions. TRAIL, a killer cytokine. The ability of the cells to resist apoptosis by TRAIL, apoptosis measured here by activated caspase-3, is diminished, more cleavage of caspase-3, more apoptosis when we deprive them of SARC. So this set of findings led us to consider this notion that on arrival in the bone marrow, circulating breast cancer cells that have a robust level of SARC, and we don't know molecularly exactly what we mean by that. SARC is there, the SARC gene expression profile is there. How is SARC activated? Are there other signals? Are there alterations in SARC or its regulators? However, it happens, SARC enables these cells to respond very well to something that is particularly enriched for in the bone marrow and already known to be playing a role at the level of the CXCR4 receptor from earlier studies, in addition to providing defense against something that is also in the bone marrow, and that would be deleterious, contributing, we think, at least partly, to allowing these cells to be a competent yet latent disease, awaiting their chance for full evolution towards a bone metastatic phenotype, and as Klaus indicated, also quite likely be poised to serve as seeds for metastases, infiltration at least, to other sites. This, for consideration, raises the issue that SARC inhibitors might, perhaps even transiently for only a short limited period of time, might be a meritorious adjuvant therapy in ER-positive patients, as well as SARC-positive, ER-negative breast cancer patients, to eliminate from their marrow disseminated cells. I want to close by indicating that this story pertains to breast cancer. We need not think that, we know that not all cancers or all metastatic processes have a built-in long late metastatic latency process. Some of them do not. Lung adenocarcinoma is not years, not decades, it's typically weeks to months. In this case, the questions must be different. Not so much about what confers latency, but what is that confers these cells with the ability to metastasize forcefully soon after the tumor is releasing them. And just to highlight our recent progress on this, similar approaches have allowed Don Nguyen to identify that in the case of lung adenocarcinoma, it's a hyperactive Wnt pathway that endows these cells with this fuller ability to metastasize aggressively to lung and to bone. So I'm contrasting the course of metastasis like I'm contrasting the sites of metastasis between different diseases. And with this, I would like to thank the colleagues who have done this work. They are listed here. I mentioned them as I went along. And I'm very pleased to be, I guess, the closing act of this event. Thank you.
Well, thank you, Joan. Actually, a special thanks to Joan for making such an effort to be with us today.